I'm Darren, and we work on fruit flies. This is Connor.
I'm Connor.
Connor actually does all the work.
Yeah.
So Connor's a PhD student. I was a PhD student many years ago. But no, Connor is in my lab. We work on fruit flies, and we're interested in how nervous systems are built, which is, you know, essentially what we do in this department. So flies have been used for about 100 years or more, and because they've been used for such a long time, there's lots of tools and resources that are available for us to use. They go from being an egg to making an egg in about 12 days. So they've got a really rapid turnover. So we're able to use genetics to do our work, to look at the role that genes play in building nervous systems. Okay. Yeah, so we use genetics to try to understand how nervous systems are built. And we also use a lot of fluorescent proteins that have been, you know, found in jellyfish and corals, and we use those to help us to help us see cells growing. So that's what we do, and we're interested in how trees like these here, these particular neuron trees, how they're built. And that's one side of the lab. The other side of the lab is focused on how the components for the nervous system are generated, and that's what Connor does.
So, like Darren was saying, once a neuron is born, it has to make this lovely complex tree structure where it connects up to all of the things that it wants to be able to connect up to. But as well as that being an important part of generating a nervous system that functions in the correct way, another aspect of that is generating the right numbers of these cells. And that's kind of what I'm interested in. So one way in which many organisms do this is they select certain cells to be removed by something called programmed cell death. So a cell can be born, but it can be decided that that isn't going to be used in the final nervous system, and it can be killed off. And it just so happens that Drosophila do an awful lot of this during the building of their nervous system. So it's the perfect model for understanding how this form of decision making of do cells live, do cells die, instructs the formation of this complex final nervous system in the end. And that's what my project is ultimately about, understanding the how and the why, I guess, of this process of cell death in the developing nervous system. Part of what we, every experiment essentially that we do involves the flies, which we'll show you in our designated fly room, but also dissections, I guess.
Yeah.
We spend a lot of time
a lot of time
out
at these
nervous
scopes
systems.
here. So these are what we call stereo microscopes, and they're we basically take the flies that we're interested in, and we'll dissect out their nervous system. So the flies are very small, as you can see, tiny little flies here. So the flies are really small, and we use very fine forceps to take out their nervous system. So we'll knock out a fly and then put it in here and dissect it in this dish, which has got like a little bouncy rubber bottom to it. We'll do that in some physiological. I don't know if you can focus in on that.
Yeah, okay.
Yeah, but that's what we'll dissect the nervous system out of quite a lot. And in this case, we'll drop it into some saline, and then use these tiny little scissors here. These are scissors that are used for eye operations. Usually they're iridectomy scissors, and you can see they're really small, and we'll dissect out, we'll use it to open up this maggot and take its nervous system out.
Wow. Can you just poke it around a bit again?
Sure. So that's an L3. That's a large final install larva before it turns into a pupa, which is what we have, or a puparium and then a pupa. So that's, you can see that's started to go into metamorphosis there, and that will then turn into a fly. And this is, we've done a lot of work on how the nervous system changes during metamorphosis. That's what we basically study that time.
What's the stuff at the bottom of that?
This,
Yeah.
this is food. This is just a yeast and agar paste. The flies find delicious. So, so these are a different type of fly. They're called the black soldier fly, and they're being used as a protein source. And it's a new way of sort of getting rid of waste. And there's a company around the corner, just under London Bridge train station, that grows these, and they make protein powder out of it. So maybe in the future we'll be eating protein from these creatures. So
cool. So yeah, Darren was just talking about all of the dissections and experiments we do out in the bench space, but the majority of the time when you're working with flies, you're actually going to be spending it in here, which is what we call the fly room, conveniently. And this is where everybody, all of the people working within the various labs that are studying using Drosophila, we all keep our flies in here basically. So all of these racks on the walls, all over here, these are mine over here, contain all of the fly stocks that we're working with. And so these will be flies that have different genes knocked out, so removed, for example, or they might have fluorescent proteins like the ones Darren was talking about earlier, shoved into genes so that we can see where those genes are, when they're on, and
how do you
what
do
they're
that?
doing. So you can do that by essentially the wonders of cloning, and the fact that all organisms use a common blueprint for making proteins, basically, so that's DNA. So as we spoke about before, what people have done in the past is take regions of DNA from jellyfish, for example, which codes for a protein that is fluorescent, so you can see it glows, and then inserted that by injecting DNA into the fruit flies and seeing if it lands inside the genes. And in doing so, have made a version of that gene whereby when that gene is made, the product glows or is fluorescent and we can see it. So it's a crazy tool that we use every day, and it's kind of integral to every model organism or every lab that is currently working in any form of biology, really. We use it rather extensively in Drosophila because it's so, I guess historically, it's been an organism where lots of these sort of tools have been generated. So yeah, all of the flies are here. We have these two great big incubators in the corner, and these are kept at a stable 25 degrees, which is kind of like the optimal temperature that you can culture these Drosophila at. And there is a little window, and you can probably just see these are everyone's stocks that they're trying to keep at that optimal temperature and get growing at the perfect rate. So I think mine are on the top shelf, and then everybody else's are going down. And these are incredibly important for basically keeping our flies alive and keeping them developing at this really tight, regulated fashion. Yeah, so each of these microscopes is linked up to a carbon dioxide pad.
What does the carbon dioxide do?
And the carbon dioxide anaesthetises the flies, so it sends them to sleep. So if you want to work with them, obviously they will fly away unless they're asleep, which is one of the issues that we have.
Mm-hmm.
So if you want to collect a certain type of fly, you pour them out onto this pad, the CO2 knocks them out, and then you can actually go in with a paintbrush, which is what we typically use, and sort out the flies. You might be looking for male flies, or you might be looking for female flies, or flies that have one of those genes I mentioned earlier, or flies that don't have that gene. And we would do that all underneath these microscopes. with the flies asleep on the pad.
Okay, fantastic.